Thank you, Misha, very much. Thank you for the kind words, invitation. It's great to be here, and I visited your fantastic city yesterday, so first time, so I really enjoyed it a lot. Thanks, Sasha, for that. Okay, so my lab in Strasbourg is interested in blood platelets since a very long time. And platelets, you're probably all familiar with platelets, a small circulating cell in the blood. And it has this typical discoid shape that you can see here. I would like to show you this quick video from Anita Ekley in the lab using a dual beam microscope, if the video works. So these are a section of platelets, and she can, with the electron microscope, with the dual beam, go through this section and finally reconstruct platelets. And this is actually a very nice tool because it gives us some insight in how platelets are organized and information about the inner organoids, how the organoids are organized in the platelets. And organoids in the platelets, organelles are very important because they play major functions in platelets, function related to their main role, hemostasis, but also a non-hemostatic function, releasing many biologically active substances. So platelets play mainly a role in hemostasis, which are the mechanisms leading to the arrest of bleeding. After vessel wall injury, you have the adhesive proteins that will be exposed in the vessel wall. Platelets will rapidly stick to them. They then become activated and they lose their discoid shape, so that this discoid shape that you can see on the top is lost. The platelets contract and they start to extend filopodia. Activation also leads to platelet aggregation and formation of a plug. Then you have the coagulation cascade that is activated, thrombin is generated, fibrin is formed that stabilizes the clot and will then restore normal blood flow. So the mechanisms involved in the different steps of the formation of a plug are actually quite well known, and I've represented some of them here on this slide. And what we know today is that on the condition of elevated blood flow, as you can find in arteries or in microcirculation, the first step of platelet recruitment at site of injury is ensured by the GP1-B59 complex, which will bind to VWF, that is in the subendothelium. You then have stable adhesions to integrins, mainly of the beta-1 and beta-3 family. You have actually 3 beta-1 integrins, alpha-2, alpha-5, and alpha-6 beta-1, which bind respectively to collagen, fibronectin, and to laminins. So this stable adhesion is then important to ensure interaction between some platelet receptors and proteins in the layer, such as collagen. So you have this GP6 collagen interaction, and this really initiates signal together with the integrins that will lead to an initial phase of platelet activation. They will change their shape, but also release many soluble activators, including thromboxane A2, ADP, ATP. And these soluble mediators that are also called the amplification loops are actually very important in further activating the platelets. So they can act back on the same platelets or on many other platelets, circulating platelets, for instance, further activating them. And notably, they will increase the affinity of integrin alpha-2 beta-3 for its main ligand, fibrinogen, and then support platelet aggregation and finally form the hemostatic plug to stop the bleeding. So this mechanism taking place in physiology can also take place under pathological settings, notably in diseased vessels presenting a thickening of the wall due to atherosclerotic plaque formation. And erosion or rupture of such a plaque will promote the adhesion, activation, and aggregation of platelets. But under this condition, because the plaque material is believed to be very reactive, but also because the blood flow conditions are profoundly modified due to the stenosis, platelet aggregation, that this time is called thrombus formation, can become occlusive. And this will induce life-threatening ischemic pathologies such as myocardial infarction and stroke. In the last 20-30 years, several antiplatelet agents were developed. The first one was aspirin, that inhibits the synthesis of thromboxane A2. You also have blockers of the ADP receptors, The best known is clopidogrel, but there are some new ones, such as prasugrel, for instance. And then you have the blockers of integrin alpha-2 beta-3. One example is apiximab. So these drugs have proven efficacy in both the treatment and prevention of ischemic events, but they also have limits. And one of them is the bleeding risk. And on this slide, it's actually interesting to see what they target. You see that they target either the final stage of platelet aggregation or the intermediate stage of platelet activation, which is the case of aspirin and clopidogrel, but none of them is actually targeting the initial phase of platelet adhesion, so in the interaction of platelets with the surface. And in the lab, we are notably interested, at least one of the team is interested in better understanding the interaction of platelets with subendothelial proteins, in order to understand really the function of these receptors, but also to determine whether they could represent potential antiplatelet targets. And today, I will focus on one of these interactions between α6β1 and laminins. So first, what are laminins? Laminins are a component of the basement membrane that just lies underneath the endothelial cell layer. And this basement membrane is composed of a sheet of proteins in which you find, of course, lamins, but also perlecan, type 4 collagen, and tactin. Lamins are cross-shaped heterotromic proteins composed of an alpha, beta, and gamma chain. The alpha is particularly important because that's the one that is going to interact with proteoglycans, but also with integrins, so interact with cells. Lamins are usually called according to the chain numbers, so that laminin with an alpha 1, beta 1, gamma 1 chain is called laminin 111. So what do we know about laminins in the vessel wall? Actually, they have three different types of laminins. The one with an alpha 5 chain, so 511 and 521, with an alpha 4 chain, 411, 421, and with an alpha 2 chain, 211 and 221. And this is something, when we started, we wanted to control. So what we did is immunofluorescence on mouse carotid arteries. We used antibodies against the various alpha chains of laminins. We did not see any signal with alpha 1, an antibody against laminin alpha 1. We had some signals with an anti-alpha 2 chain laminin antibody, and also clear signals with antibodies against alpha 4 and alpha 5 chains. So it clearly indicating that indeed you find alpha 2, alpha 4, alpha 5 chain containing laminin, but not alpha 1 in at least this type of vessels. So then we wondered, okay, what is known about platelet interaction with laminin? Actually, when we started this work, there were already several publications out there. And what is quite clear from this paper is that if you immobilize the laminins onto a surface, and then wash platelets on the top of them, they will adhere and they will become activated. And you can see on these images that they nicely spread over the surface. And this process clearly relies on integrin α6β1, since if you use a blocking antibody, you completely get rid of the platelet laminin interaction. When we then looked a bit closer at the studies, what we observed is that most of them were using laminin 111, and 2 of them were using placental laminins, which is composed of various types of laminin, but you mainly find also alpha 1 chain containing laminins. So these experiments were actually performed with non-vascular laminins, and we wondered what the interaction between platelets and vascular laminins are. And for this, we decided to use a flow system that you also use here, in which anticoagulated blood, so we use hirudin here as an anticoagulant, is perfused through a flow system that we then coat with purified proteins by using a syringe pump. And here is a video of what you see when you perfuse blood over laminin. You see mainly individual platelets sticking down, and they over a while cover the surface. So there's no aggregation going on, it's mainly a platelet, single platelet adhesion. So we then decided to look at all the different vascular laminins, compare them to laminin 111, because that's the one that was studied in literature, but also to 2 well-known adhesive proteins for platelets, fibrinogen and VWF. And these experiments were performed at a wall shear rate of 1,000 per second. We did not see any platelet adherence adhering on laminin 211. We had nice adhesion on laminin 111 and 411 that was quite equivalent to what we had on fibrinogen. And we were very impressed with the adhesion we observed on α5 change containing laminins, 511 and 521, that was even more impressive than on VWF. So that's the picture you have at 1,000 milliseconds, and we next wondered what would happen at much lower shear rate that you can have in large arteries, in veins, or at even higher shear rates that you find in microcirculation or under pathological condition. And there you have the result in which we express the number of platelets as a function of wall shear rate. So what comes out of this is that whatever the wall shear rate you use, there is no adhesion on laminin 211. 111 and 411 were quite adhesive with still nice adhesion at 1,500 milliseconds. And again, we're quite impressed with the adhesion that we observed on laminin containing α5 chain with significant adhesion at 3,000 and 4,000 milliseconds. So now, what about the molecular mechanism involved in the interaction? And we first focused on the beta, on the integrins, and about the beta-3 integrin. So we used here ReoPro and observed that it does not impair platelet-laminin interaction as you would expect, at least on laminin 411 and 511. And we confirmed that blocking either α6 chain or beta-1 chain completely prevented platelet interaction with the laminins. So because we had this interaction and adhesion at very high shear, and because it is well accepted that the only platelet receptor supporting platelet adhesion on the high shear is the GP1 benign complex, we looked at its role here, and here at the wall shear rate of 1,500 milliseconds on the 3 vascular laminins. And what we observe is that blocking the A1 domain of VWF, so binding domain on GP1B, or at the opposite, GP1 binding domain on VWF, inhibited quite strongly platelet interaction on all 3 types of laminins, clearly indicating that indeed the GP1B VWF axis is important here. And to further show that a role for VWF, we used blood from VWF deficient mice, perfused it over the 3 laminins, and as you can see here on the bottom, we had marked inhibition in platelet recruitment on the laminins. So how does this work? We hypothesized that it might be like with many other adhesive proteins, so an interaction between VWF and laminins. So we actually went back to a purified system, immobilized laminins onto the surface and added VWF on the top of it, and did this the other way around as well. So immobilize the various immerse VWF and add laminin, or immerse laminin and add VWF. And in each cases, we saw significant interaction between both. So VWF is indeed able to stick to the various vascular laminins. Then we move to evaluate platelet activation. So here it's after perfusing blood over laminins. We fixed the platelets and looked at them by SEM. And what you can see here is that almost all the platelets adhering to this different type of laminins become activated. We had the feeling that the laminins with the α5 change, the platelets were more activated with not only phenopodia but also lamellopodia. So to better define that, actually what we did is we used the dual dye calcium assay. So here what you do is you isolate the platelets, you treat them with calcium dyes, here green Bapta IM, and you add them back to washed red blood cell and perfuse this over you immobilized laminins. And so what you see is that when the platelets are resting, they will appear in blue, and then they become purple, red, or orange when there is some activation. And this is what we observe. You can see that the platelets adhering start to blink and go from blue to purple, and even sometimes you have some red going on. So once you have this type of videos, you then focus on single platelets and extract a calcium profile. And what we see is that we have mainly calcium spikes. And then you can do this on a population of platelets, and indeed what you observe is that the α5 chain containing laminins were more reactive, had a higher calcium response than the other ones. So how can we explain that? Actually, there was a paper from the group of Steve Watson that suggested that glycoprotein 6 is involved in platelet laminin interaction. So this is something we looked at, especially because we're interested in this receptor. And we used blood from JP6 deficient mice, perfused them over the various laminins. And on the left side, we evaluated the adhesion. There's no difference whatsoever. And on the right side, we actually evaluated the calcium response. And what we observed is that in the JP6 deficient platelet, we had a reduction in calcium signaling on the alpha-5 chain laminins. So that potentially explains the difference we have between the laminins. So in conclusion of this part, this result suggests that vascular laminins support efficient platelet adhesion and activation under various blood flow conditions. So the next step for us was then to evaluate the importance of this platelet-laminin interaction. And to do so, we generated a mouse that does not express α6β1 in platelet using the Pьер Krelok system. So we used mice that express Krelok on the promoter of Pьер that is believed to be platelet specific, even if this we can discuss. And we crossed them to mice having the α6 gene gene for α6. So the mice we generated are actually viable and fertile. They have a normal platelet count. They have a normal expression of all the known platelet receptor, apart, as you would expect, of integrin α6, which was very markedly reduced. So we first characterized these mice, performed some aggregation assays, and as you can see here, so these are with washed platelets. Alpha-6 deficient platelets respond normally to a series of agonists, including ADP, thrombin, pathopeptide, thromboxane 2 analog, collagen, CRP, which is a specific agonist for JP6. They had also normal responses to fibrogen binding in response to ADP, thrombin, pathopeptide. P-selecting exposure was completely normal to soluble agonists, as well as annexin 5 binding. What about the ability to adhere to various surfaces, and first of all, laminins? We are pleased to see that when we perfused hydrogenated blood over vascular laminins, there was almost no platelet interaction with the surface anymore. So confirming that in the mouse system, α6β1 is really playing a key role in platelet laminin interaction. This is shown as well in the quantifications. And as a control, we looked at various adhesive proteins. Von Willebrand factor, we had not much of a difference. Fibrinogen, no difference. Fibronectin, there was a little bit something, but nothing significant. However, we were a bit surprised to see that there was a reduction when we perfused blood over thrombosporin, and this was actually significant. So α6β1 must probably interact with thrombosporin. Okay, so after this characterization, we went then back and to the main question, which is, what is the importance of α6β1 in thrombus formation? And for this, we developed a model, a mechanical model, inducing an injury in the carotid artery with a wire. And what you do here is you insert the wire in the carotid artery, move it back and forth a couple of times. This model was characterized. This you can see on the electron microscopy images. You can see the endothelial cells, noted EC here in the vessel, in the control. And once we use the guide wire, after moving back and forth, the endothelial cell layer was removed, and you start to have to see platelets adhering. And actually, this you can do in real time. By intravital microscopy, and in black is the profile you have for white-type platelets, so biphasic thrombus formation, and when we use the α6 knockout, we had a marked decrease in thrombus formation. We moved then to another model, again mechanical injury, this time with a forceps, but of the abdominal aorta. This model was also characterized, and when we actually pinch the aorta, we remove the endothelial cells, and a thrombus is starting to form. And this also you can follow by intravital microscopy. You have again a reversible thrombus formation as often, and this thrombus formation is reduced both in α6 deficient mice but also in β1 knockouts. And finally, we used a laser injury model of mesenteric arterioles, So the specificity of our lab, of our model, is that we have 2 degrees of severity. This model were characterized, I don't show the images, but just schematics to explain a bit. So when what we call the superficial laser injury, we basically remove the endothelial cell layer. And we also have a model of severe laser injury where we really expose the inner layers of the vessel. And what we observe is that there was a marked reduction in thrombus formation after superficial injury, but not much of an inhibition after a deeper injury, which makes sense because you're not exposing much of the basement membrane here, so not much laminin is exposed. We looked at hemostasis using a tail bleeding assay. So what you do here is you savor the tail of the mouse, inject it, put it into saline, and measure the time of the arrest of bleeding. And we did not observe any increase in bleeding time and no increase in the volume of blood loss. So in summary of this first part, what this result tends to suggest is that after vessel wall injury, laminins are exposed to the flowing blood. They might adsorb VWF, thereby recruit circulating platelets through the GP1B9 complex, so here GP1B alpha. Then there would be some stable adhesions through α6β1 that binds to the laminins, but potentially also to thrombospondin 1, facilitating the interaction between GP6 and the α5 chain of the laminins. All this receptor then can integrate signals that will lead to increase of intracellular calcium levels and platelet activation. This result also suggests that deficiency in α6β1 does not impair hemostasis, and that integrin α6β1 plays an important role in thrombus formation after superficial arterial injury, but not after severe injury. So the question here is, so what now? Where do we go? Is this any important for arterial thrombus formation? Because you think that arterial thrombus formation might actually be more severe injury. Actually, not necessarily. You do have some events of erosion of plaque that could expose, just the basement membrane. And then I would like to show a piece of data from a group that has been published. And it's quite interesting because what it shows is that if in normal vessel you do have laminins just, in the basement membrane, in atherosclerotic plaques, you have it all over the plaque. So which means that after a severe injury, you might have lots of laminin exposed, and they even observed that in this. So they've shown this in mouse and human plaques, and they also show that there is increase in expression of alpha-5 laminin in the APOE deficient mice. So it's a mouse model of atherosclerosis. Okay, so now I move to the second part of the talk, and here I would like to shift a little bit. So we know that platelets play a main role in hemostasis and arterial thrombosis, but they are also involved in many additional processes, some physiological processes, such as innate and adaptive immunity, tissue repair, angiogenesis, but also in pathology, inflammation, atherosclerosis, and in cancer, notably in metastasis. And we got interested in their role in tumor metastasis. So what is known about that? Actually, almost 50 years ago, there was a publication from Gabriel Gazik, who showed that platelets, he was the first to show that platelets play a role in metastasis. And what he did, he actually treated mice with neuraminidase to induce a thrombocytopenia, and then injected tumor cells into the tail vein of these mice and looked at metastasis after 15-20 days in the lung. So it's colonization of tumor cells in the lung, and he observed that they were markedly decreased. So this result has been confirmed by many groups, and this slide just shows a view of potential roles of platelets in metastasis. So what do we know? We know that when a tumor cell detaches from a primary tumor and enters the blood circulation, it will interact with platelets. This interaction clearly leads to platelet activation. Activated platelets will then release the granule content, and you have many biologically active elements, notably TGF-beta, which can then act back on tumor cell and promote EMT, so help them to become more invasive. It has also been proposed that platelets would form a physical shield around the tumor cells and protect them from the effect of the shear forces, but also from the immune system. What has also been proposed is that tumor cells in the vessel could promote some inflammation. We know that inflamed endothelial cells can release VWF, and that platelets can then interact with it. So there is the idea, still needs to be confirmed, that platelets could facilitate the recruitment of tumor cells at distant sites. And finally, it has been proposed, it's known that platelets regulate vascular integrity, and it has been proposed that they might help the tumor cell to extravasate. So that's a bit the view that there is today, but what is still lacking is the molecular events that are involved in this process. So what do we know about platelets and the platelet receptor? Actually not much. There are several studies suggesting, actually only one paper suggesting that glycoprotein 6 and CLEG2 could be involved, so this still needs to be confirmed. There has been paper, but there is controversy on the role of the GP1B59 complex and integrin alpha-2 beta-3 with really opposite results. So we're not too sure what these receptors are doing right now. And actually concerning the beta-1 integrins, there's nothing there. So we decided to have a look at the beta-1 integrins and focus a little bit on alpha-6 beta-1. And for this, we used an experimental metastasis model, which consists in injecting B16F10 cells that express luciferase into alpha-6 and beta-1 deficient mice, and follow over time the formation of metastatic nodules into the lungs by using bioluminescence system. And as you can see on these images, we had less signal in alpha-6 deficient mice and beta-1 deficient mice as compared to the controls. And this was observed, you can see on the quantification, at different stages. At endpoint, we also, so after 20 days, we also isolated the organs and looked them with this device and observed that the lungs were indeed, there was less signal in alpha-6 and beta-1 deficient lungs as compared to the controls. And we also did some staining, some histology. In parallel, what we did is a more classical method. We used the B16F10, injected them to the tumor cell and to the mice, and looked at the presence of nodules, which are the black dots that you can see here, on the top of the lungs after 15 days. And you can see on these images that indeed there's a reduction in formation of metastatic nodules on these lungs. So is this only occurring with this specific tumor cell line, or is this more broad? To answer that question, we actually use different types of tumor cells: MC38 colon carcinoma cells, E0771 medullary mammary adenocarcinoma cells, and 83 breast cancer cells. They were all injected in the cell vein, and after 20 days, we did some histology on the lungs. And you can see on these bar graphs that for all three types of tumor cells, we had a reduction in the number of lung metastases. We then moved to a model that might be a bit more relevant. This is a spontaneous model. So in this model, we used 83 cells, so that are isolated from an MMT-PYMT transgenic mice, and they are injected in the mammary fat pad of 8 weeks old female mice. And what is interesting in this model is that you will form the primary tumors that will then spontaneously metastasize in the lung. So what we first did is we followed the tumor growth over time, and we observed no difference between alpha-6, beta-1 deficient mice and their control. So no effect on primary tumor growth. However, when we looked at lung metastasis, we observed a clear decrease in tumor metastasis in the alpha-6 deficient mice by Giemsa staining or by hematoxylin eosin staining. This was significant. So is this really platelet related? We use a PF4 Krelok system, but we could argue there that maybe it's not as specific. And to do that, we actually did an experiment in which we transfused platelets. So the idea here was to use a mouse that has a low platelet count. so knockout for MPL, the thrombopoietin receptor, and these mice have actually 75 to 80% reduction in platelet count. When you then inject tumor cells in these mice, you do have a marked decrease in the number of lung metastases. And what we did is that while we injected the cancer cells, we transfused platelets. And when we transfuse white type platelets, we actually are partially restoring the metastatic process. But this we did not observe when we transfused α6 deficient platelets. So what is the mechanism? Obviously, α6β1 is an integrin, surface receptor, could be involved in the platelet tumor cell interaction. This was the hypothesis. So what we did is we used tumor cells, isolated platelets, let them interact, and these are SEM images on the left side. You can see many platelets interacting with the tumor cells, and when we used α6 deficient platelets, we observed less interaction. This is actually not easy to quantify, so we developed an assay, a static adhesion assay, in which we used washed murine platelets deficient for either α6 or β1 integrin, or washed human platelets with blocking antibodies against α6 or β1. The platelets were fluorescently labeled here in green and added on the top of murine or human tumor cells. And on the right side you have an image. And what we observe is less signal when we used α6 deficient platelets as compared to white type platelets on 83 mammary carcinoma cells. This was confirmed for mouse platelets on different types of tumor cells, B16F10, 83 and MC38, both in the α6 deficient mice and in the β1 deficient platelets, so sorry, not mice, platelets here, it's a purified system, but also with human platelets on different types of tumor cells, MDAMB231, SKRB3 and Novo cells, in the presence this time of washed platelets with a blocking antibody against either α6 or β1. So that's what we observe in vitro. Do we have a chance to evaluate this in vivo? We tried. We co-injected in a mouse fluorescently labeled, so CFSC 83 cells, together with an antibody, homemade antibody RAM1 that specifically recognizes platelets. After an hour, we euthanized the mice, collected the lungs, and did some immunostaining, detected some green signals. Actually, the tumor cells are going within 15 minutes, you have all the tumor cells in the lung, and looked at the red signal in contact with the green one, quantified it, and observed that there might be indeed a decrease in this platelet tumor cell interaction. So what might be the control receptor? We hypothesized it might be ADM9, which is a disintegrin and metalloproteinase 9. It's a multi-domain type 1 transmembrane protein composed of many sites. One of the regions that is of interest to us is the disintegrin region, because it has been proposed to interact with integrins. So ADM9 is known to release biologically active ligands and to promote cell adhesions through the binding of integrins, and notably α6β1, and that's why we picked ADM9. So it's involved in various diseases, rheumatoid arthritis, Alzheimer's disease, atherosclerosis, asthma and cancer. And concerning cancer, it has been shown to be overexpressed in melanoma, breast, colon, ovary cancer, to be correlated with cancer progression, and there is a bad clinical outcome linked to the expression. So to evaluate the role of ADM9, we first generated tumor cells that do not express anymore ADM9 using an shRNA approach. And we generated two clones for two cell lines, the 83 breast cancer cells and the MC38 colon carcinoma cells. And we checked that there was less expression by mRNA, but also at the protein level by Western blotting and immunofluorescence. And then these cells, we use them in the in vitro platelet tumor cell adhesion assay. And what we observed is that the two clones of the 83 cells reduced by approximately 50% the ability of white type platelets to interact with these clones, and this level of inhibition, of reduction, was quite similar to what we had with control cells in the presence of α6 deficient 1 platelets. And interestingly, when you combine deficiency of α6 beta 1 on platelets and ADM9 on tumor cells, you don't have any additional effect, suggesting that they could potentially interact. And we had very similar results with MC38 tumor cells. We did some experiments in a purified system to show that platelet α6 beta 1 could potentially bind to ADM9. We actually used the peptide that covers two domains, the cysteine-rich domain and the disintegrin domain that binds to integrins, immobilized it on the surface, added washed platelets, and we observed adhesion of white type platelets with a reduction when we used α6 deficient platelets. We also did this under flow conditions. We observed some interaction of platelets on the DC9 peptide that was again reduced when we used blood from α6 deficient mice. Then we went back to the in vivo model, and first the spontaneous metastasis model. I injected the different type of cells in white type or α6 deficient mice. And what we observe is whatever the condition is, there is no impact whatsoever on the tumor growth. However, we do see a reduction in lung metastasis. And again, here it's very similar to what we had in the adhesion assay. There is a reduction for the two clones when they were injected in white type mice, a reduction in the number of metastases, similar to what we had when we injected white type control cells in α6 deficient mice, and no further reduction when combined deficiency in α6 beta 1 mice and when they were injected with control in tumor cells devoid of ADM9. And we had very similar results in the experimental metastasis model with both 83 cells but also MC38 cells. So then we wondered what this interaction through α6 beta 1 and ADM9 is important for, and wondered whether this could be important for the extravasation step. And for this we used the Boyden chamber. We added tumor cells in the upper compartment in the presence or absence of platelets, and this one was covered with endothelial cells, and looked at their ability to transmigrate. And what we observe is that when you actually, so second bar, when you actually add platelets to the tumor cells, you markedly increase this transmigration. And when instead of white type platelets you add α6 deficient platelets, you lose this transmigration. And we had similar results, so no rescue when we actually used cells that do not express ADM9. Can this also be confirmed in vivo? For this, we injected 83 cells in α6 beta 1 deficient mice, white type mice. And after a couple of hours, we euthanized the mice, collected the lungs, and had a look and checked whether the tumor cells were intravascular or extravascular, and quantified that. And we actually had an increase in, a reduction in the number of tumor cells that extravasated in the α6 deficient mice as compared to control. I will finish by 2-3 slides on the pharmacological approach, because all we did for now is using knockouts. So can we also confirm these results by using an antibody? And we used GOH3, which is a known anti-alpha-6 blocking antibody. We first checked that it did not induce any thrombocytopenia. And what we observed is that when we co-injected this antibody with 83 cells and looked after 15 days at the number of lung metastases, we indeed observed the reduction. And this reduction, there was no effect when we injected GOH3 in alpha-6 deficient mice, as you would expect, because there is no target there. And we actually were able to confirm this result by using B16F10 cells as a model. So in summary of the second part of my presentation, is this work shows that platelets and integrin α6β1 can mediate platelet tumor cell interaction through the binding of ADM9 on tumor cell, and that this interaction is probably important, at least for extravasation. So in conclusion, and this is something we are really interested in the lab, we believe that targeting the adhesive property of platelets, talking about, I showed data today concerning beta-1 integrins, but it might also be true for the GP1 beta-9 complex, and for sure it's true for glycoprotein 6, might actually represent a safe anti-thrombotic strategy. And we have ongoing work on JP6. JP6 is very interesting because it's only expressed in platelets, so it makes it very relevant, and it plays only a minor role in hemostasis. And then targeting this adhesive properties on platelet may also reduce platelet interaction with circulating tumor cells and potentially reduce tumor metastasis. And with this, I would like to finish and just thank a couple of people from the lab. Christian Gachet, who is the head at our facility, Elmina Mamadou-Wabak, who has done all the second part of the story I mentioned today, and Mathieu Schaff, who has done all the first parts on α6β1 and thrombosis. And I would like to thank some collaborators involved in this work, Elisabeth Georges-Labouesse for the mice, Scott Abrams for the 83 cells, and Paula Zigrino for the ADM9 tools. And with this, I would like to thank you for your attention.